Medical bioinformatics methodology and workflows constitute a structured framework for transforming biological and clinical data into knowledge applicable in the healthcare field. In this sense, medical bioinformatics as an interdisciplinary field integrates biology, medicine, and computer science to analyze genomic, proteomic, metabolic, and clinical data using algorithms and computational models. The process begins with data acquisition from multiple sources, such as DNA sequencing, electronic health records, or medical images, and continues with pre-processing focused on error correction, noise removal, and normalization. Subsequently, exploratory and descriptive analysis is performed to identify patterns, trends, and relationships, incorporating statistical models and machine learning techniques that allow for the construction of predictive and, in more advanced stages, prescriptive models. From an operational perspective, the creation of workflows requires integrating sequential phases of data collection, aggregation, analysis, and interpretation tailored to the specific objectives of the study. In this context, application programming interfaces, APIs, enable communication between systems and facilitate interoperability between platforms, while languages such as R and Python offer specialized libraries for processing and modeling biomedical data. Taken together, a well-structured bioinformatics workflow ensures analytical efficiency, data quality, and methodological rigor, establishing itself as an essential component of precision medicine and translational research.